Hello, my name is Marcus Morris.
And I'm Samuel Berger.
And we are research students at the STEM-certified Academy of Science Research and Medicine and are currently conducting research on algal biofuels. Throughout our project, we will be utilizing the engineering design process of ask, imagine, plan, create, and improve.
One of the biggest threats to both humans and the environment is rising global temperatures. As greenhouse gas emissions continue to increase, more and more heat is trapped by the atmosphere. Alternative energy sources are necessary to protect the environment. Most sources either require an incredible amount of upfront investment or use up otherwise valuable resources, or both. A solution that wouldn't change the infrastructure of the country to a great degree would be the use of biofuels. These can use much of the same infrastructure as traditional fossil fuels, but first and second generation biofuels are either expensive to produce or sacrifice another essential part of American production, such as agriculture. Algae are promising alternatives that can be grown in seas where nothing else can be produced or in non-arable land. However, the procedure to extract the needed fats from algae is expensive and complicated. This is the problem that we hope to resolve. During our preliminary research, we discovered that by neutralizing a part of the DNA of the algae Chlamydomonas reinhardti, we could increase the production of the fats needed to create biofuels to the point where they are freely ejected by the algae.
Before starting the project, we developed a general overview of our research to follow throughout. As recommended by our qualified scientist, Mr. Pedersen, we ordered and received materials through VWR International and Aegean. While we waited for our order containing our microalgae and plasmid to arrive, we began designing our artificial miRNA using the web designer, WMD3. Once the algae gets here and we make the miRNA, we will genetically modify the algae using the heat shock method and reculture the now transformed algae. Afterwards, we would analyze our results by using a combination of qualitative observations along with PCR and fluorimetry. We used a graphing composition book to record our data, observations, and detail our methods used. We took notes of our research online using Google Docs so we can save the links for later use when publishing our paper in the Journal of Emerging Investigators. The results would lead to a proof of concept that miRNA could be used to silence genes in other species of microalgae in the same genus. So far, we have only gotten our algae cultured and purified our plasmid. We are ready to make our miRNA and transform our algae soon after.
In the beginning stages of our research, there were several obstacles we encountered and how to overcome. Initially, we wanted to conduct research on kudzu use as a biofuel, as we'll continue with past year's research and address an issue more relevant to Georgia. Unfortunately, we ran into problems with how to actually apply the research in terms of funding and the actual materials and methods. So instead, we decided to shift our focus into a field that we had read about a year prior and seemed to be much easier to accomplish: algal genetic engineering. So far, we have purchased both the plasmids and oligos that we require to silence CRAX1 and increase productions of lipids. Our next steps are to introduce the oligos into our plasmids, introduce those plasmids into our algae, and then analyze the result by comparing the lipid content in a control source of algae to our experimental genetically modified algae. Throughout our research project, we have repeatedly roadblocks that could have been avoided if we had spent more time rereading our literature. The biggest lesson that we have learned is that while at the same time it is imperative that we keep moving forward as quickly as possible with our research, we must also be well informed.